It's our pleasure to have Marcus Fandrich speak today in our ZOOMinar series, and I will just give the turn to Baron Brief to give the introduction. Welcome, both of you. Glad to see you.
Hello.
Yeah, hello, everybody. It's my pleasure and honor to introduce Marcus Fandrich from Ulm University for today's ZOOMinar. Actually, Marcus studied biology at the University of Heidelberg in Germany, but then decided that he wanted to rather move to UK when this was still easily possible and join Chris Dobson's group where he did his PhD. And there he actually looked at myoglobin and could identify that an alpha helical protein can also adopt amyloid fibrils. After his this PhD thesis, he moved back to Germany and went to Jena, where he had a short postdoctoral stay, and then finally also became a group leader. He was funded by the BMBF and had a BioFuture grant, then another independent research grant. He moved further on to Halle, and this was the second stay basically in around Gunther Fischer's institute at the Max Planck Institute in Halle. And since 2012, he's a full professor for biochemistry at Ulm University and head of the Institute for Protein Biochemistry. Since a few years, he's also coordinating a DFG-funded research center on AL amyloidosis. And I guess that he will talk about this today. And I'm very much looking forward for your presentation on ex vivo amyloid fibril characterization and determination using Cryo-EM. So, Marcus, the stage is yours. Looking forward for your presentation.
Ladies and gentlemen, dear Bernd, dear Rams, dear Magda and Sam, thank you for the kind invitation. It's a pleasure to speak in this series. So about 10 years ago, my group started a research program in which we extracted amyloid fibrils out of patient and animal tissues to make this material then available for structural analysis using Cryo-EM and other methods. And the first fibril for which we could then obtain a reasonable structure was a fibril that we purified from a mouse with systemic AA amyloidosis. This fibril consisted of two protofilaments, and the two protofilaments have a cross-beta structure. The individual strands of the beta sheets are not exactly perpendicular to the fiber main axis as was proposed in the initial models on cross-beta sheet structures, and also the individual molecules are not exactly in only one plane, but they move a little bit up and down along the z-axis, which sterically interlocks the fiber structure. The beta sheets are in all fiber structures which we determined ever since, and also here in this structure, parallel beta, so there is no anti-parallel connectivity in the individual cross beta sheets, and this fibril, as I said, has two protofilaments which are entangled in a left-handed fashion. Meanwhile, we have now obtained Cryo-EM structures from different diseases, systemic AA, AL, and ATTR amyloidosis. For those who are not so aware of it, systemic amyloidosis means that the fibril precursor is produced in one organ, and then these proteins are circulating typically in the blood and cause deposits in other sites of the body, usually in multiple organs. Systemic amyloidosis is different from localized forms of amyloidosis like Alzheimer's disease, where the organ that produces the precursor protein is also the organ where the fibrils become deposited. In my talk today, I will mainly focus on the progress which we made on systemic AA and AL amyloidosis. So systemic AA amyloidosis arises from the misfolding of serum amyloid A protein, which roughly has around 100 amino acids. It is an all-alpha helical protein that binds to high-density lipoproteins. The protein is upregulated during inflammation, and chronic inflammatory conditions like rheumatoid arthritis, leprosy, or tuberculosis are typical risk factors of systemic AA amyloidosis. In these acute phases, the peak levels of SAA are about 1,000 or 10,000-fold higher compared to normal physiological blood to reach peak concentrations of 1 to 5 milligrams per milliliter. The amyloid is deposited in spleen, kidneys, liver, where it can also cause end-stage kidney diseases, if not death, if the disease is not treated or does not go away. Current treatments are anti-inflammatory treatment to lower the serum levels of SAA. And one thing which is quite interesting about that disease is that it does not only occur in humans, but also in a large number of different animal species, birds as well as mammals, wild living animals as well as captive animals. In particular, the fact that AA amyloidosis occurs in mice gives rise to a nice model system to study systemic AA amyloidosis, and in mice, the disease also exhibits prion-like features. So if you upregulate SAA and you wait for many weeks or many months, the mice spontaneously develop AA amyloidosis, but this process is dramatically accelerated if you inject the mice with a spleen extract containing AA amyloid fibers from a diseased mouse. Then fiber formation occurs very rapidly, basically within a few days, and the mouse is full of amyloid, at least within spleen, and then also in liver and later on in kidneys. This ability to be inducible has given rise to several publications regarding the prion-like features of this disease. The fibers that can be injected are can also come from different species. There can be some type of cross-seeding, and you might be aware that AA amyloid can also be found in food products, for example, in duck or goose liver that you can commercially buy in the supermarket. You do an amyloid stain, you find AA amyloid. And if you feed such material to appropriate mice, they rapidly develop AA amyloidosis. We have obtained Cryo-EM structures from extracted amyloid fibers from different species, including mouse and human patient. The fiber structure is to some extent different in the two species. I should also say that the fiber structures which you see here are representative for the animals and for the humans, because when we extracted fibers from different mice or from different human patients with this specific disease variant, we always had the same fiber structure. Also in those cases where we had access to different tissues from the same donor, no matter whether it is an animal or a human patient, we find consistent fiber structures and fiber morphologies at different deposition sites throughout the body. The fiber protein fold is in both cases all beta and exclusively beta sheeted, no alpha helical structure, and therefore the fiber protein fold differs quite substantially from the native conformation of SAA, which is an all alpha helical protein, resembling the alpha to beta transition in infectious prions. Despite the difference in the architecture between human and the murine fibrils, there's one thing which is in common. These are the first 20 or so amino acid residues, which basically have the same conformation in both species. The rest of the molecule has a different fold, but this N-terminal arc has a rather similar structure, irrespective of whether you look at the murine and at the human protein, which perhaps suggests the partial species barrier, the kind of species barrier that exists if you inject human fibrils into mice. And you can potentially explain it with this partial complementarity of the structures. The two fibrils differ in the interface between the two protein stacks. In the mouse fibril on the left-hand side, the two protofilaments or the two protein stacks are connected via reciprocal salt bridges between basically two residues. And in the human fibril, there is an extensive packing interface between the two fibril protein stacks. It's probably not possible to really say two protofilaments because they've never been found to occur in isolation, only in this condensed structure. And there is a small steric zipper in the center. And as I said, a quite extensive packing interface between the two fibril protein stacks. Another difference is the handedness of the fibril, which you can reveal by platinum side shadowing and transmission electron microscopy or scanning electron microscopy. And in the mouse fibril, the twist is left-handed. In the human fibril, it is right-handed. And the different overall twist of the fibrils then also corresponds to the different twist of the beta sheets in the two fibrils, which is shown below. In the mouse fibril, the twist of the beta sheets is left-handed if you look at the sheet edge on. And in the human fibril, it is right-hand twisted, and therefore different from the canonical twist in globular beta sheets, which is shown in panel D, which is by this definition always left-hand twisted. So you can consider the right-handed twist of some amyloid cross beta sheets as some different type of cross beta or different type of beta sheet structure. And it is not only occurring in human systemic AA amyloidosis, we also found it in other cases, including A beta fibers, which we purified from the brain. Our structural data were informative about features of the disease. For example, there is an observation that there are certain mouse strains like CEJ mice, which are resistant to development of AA amyloidosis. Even if you upregulate SAA and you give spleen extract as a stimulus, these mice do not necessarily develop AA amyloidosis. And it was shown previously that the reason for this amyloid resistance is that these mice express specific variants of SAA protein, either MSAA 1.5 or MSAA 2.2. Now, there is in both variants 1.5 and 2.2 an exchange here at the end, which is not part of the fiber structure. In the fiber structure, there are the residues which are marked here, and the only two which are then common for both variants, 1.5 and 2.2, are these two exchanges here, an isoleucine to valine exchange at position 6. And a glycine to histidine exchange at position 7. Now, the replacement of an isoleucine by a valine basically removes one methyl group at this position, but otherwise the exchange is relatively conservative. In the case of this glycine to histidine exchange, the glycine with the alpha hydrogens points towards this hydrophobic core, which is formed by the N-terminal beta arc, which is the central and most hydrophobic structural element of the fibril protofilament. And it probably does not need a lot of imagination to see that replacement of this glycine at this position with a histidine, which then would poke into this hydrophobic pocket, is not very healthy for this fiber morphology. So we believe that the reason why these mice are resistant to development of amyloidosis is that they have a mutational change that specifically blocks the formation of one fibril morphology, of the pathogenic fibril morphology. It does not necessarily block fibril formation at all, because proteins like SAA 2.2 readily form fibrils inside the test tube. But these fibrils then probably have a different molecular structure. The fibril core is usually formed by a small part of the fibril protein. As I said, SAA has a length of about 100 amino acids, and in the mouse protein, there are about 69 amino acid residues forming the ordered core of the fibril. In the human fibril, it's only some 56 or 57 residues. Then there are a few residues which are dotted here, which are present in the fibril because we see them by mass spectrometry. We don't see them really in Cryo-EM. We see them by mass spectrometry. They are truncated at the C-terminus. The truncation is heterogeneous. The reason that we don't see that really as an ordered structure by Cryo-EM indicates that they are flexible and structurally disordered, and perhaps this diffuse blurry density, which we see to some extent around the fiber structure, could be arising from these disordered C-terminal ends. But nevertheless, there is a truncation at the C-terminus. The fast C-terminus is lost in both proteins. That was known before. And at least in systemic amyloidosis, there is an ongoing hen and egg discussion. What comes first, fibril formation or proteolysis? With these C-terminal truncations, our Cryo-EM structures would be consistent with both mechanisms, although I tend to favor that fibril formation comes first. I can explain that more in the questions if you want. But there's at least one case where we have quite some good evidence for the order of events, and that is the very first residue of human SAA protein, which is an arginine. And this residue is missing in the fibril. We know that by mass spectrometry, this residue is absent. When we look at the fibril structure, the first residue of the fibril protein is residue number 2. It is a serine. And this serine is buried deeply in the core of the fibril. Also, the alpha amino group makes contact with a neighboring carboxylate. And it is very hard to imagine that there could be first an additional arginine residue in the fibril protein, which becomes then cleaved off after fibril formation. So in this case, there's actually very strong evidence that the desargination of residue 1 must come first before the fibrils can form. A typical observation of these of our fibril samples is that they do not contain a single fibril morphology, that there is some kind of polymorphism in the mouse SAA fibrils. However, the sample is dominated by one fibril morphology, which is labeled here morphology 1. And this morphology 1 accounts for about 95% of the fibrils that are visible by electron microscopy. The remaining 5% or so are a second fibril morphology. And we were lucky enough to get also a structure of the second fibril morphology, that is the cross-section. It is very similar to the main morphology 1. It has, however, 3 protofilaments. For 2 protofilaments, PFA and PFB, the kind of interaction is very similar to morphology 1. And then there's here this additional morphology, this additional protofilament attached through 3 ionic contacts. The fibril protein fold in all cases is basically identical. Now, on the left-hand side are the two ex vivo fibril morphologies, which we extracted from mice. We then went on and fibrillated recombinant SAA protein inside the test tube, and found that we also have a sample of different fibril morphologies. In two cases, we obtained a Cryo-EM structure. And again, we find that the fibril protein fold is very similar in the different fibrils, and that there's basically a difference in the number of protein stacks, 2 in morphology I and 4 in morphology II. But the fibril protein fold is different from the ex vivo fibrils. We then went on and seeded in vitro recombinant SAA protein with fibrils which we purified from mice. Based on thioflavin T experiments, the lag time becomes shortened. These here are the unseeded fibers, which I basically showed you by the structure in the last slide. And this here is from the seeded sample. That is a Cryo-EM image of the seeded sample. And we could obtain the structure of the main fibril morphology, and that looked already very different to the ex vivo fibril that we obtained in mouse. Here is a superimposition of the two fibril structures here of the individual subunit. It's very similar. If you superimpose a stack of 10 molecules, there is a close similarity of the two structures here close to the center of the fibril. There is a little bit more discrepancy at the outside, which we believe is not an artifact or insufficient resolution, because we do actually see some differences in the 3D maps that we used for model building. But despite these small changes, we still would say that this is one fibril morphology, and that the structure of the ex vivo fibril could be proliferated inside the test tube. When we look at that a little bit more in detail, that is a slide that I already showed you, and I told you that in the ex vivo fibrils, 95% are the main fibril morphology, and some 5% are this additional fibril morphology containing 3 protofilaments. Basically, the second fibril morphology was completely lost in our seeded fibrils. We did not see it. I mean, we cannot completely exclude that it is not there, but at least it is not very abundant. And the vast majority, 98% of the fibrils that we found, is this main morphology, which corresponds to morphology 1 from the ex vivo fibrils. And these remaining some 2% or so, we could obtain an initial reconstruction, not so good resolution, but the structure is basically the one of the in vitro fibril. So in 2% of the fibrils that we see under these seeding conditions, there was a de novo nucleation reaction, and the protein adopted the fibril structure that it would adopt inside the test tube, simply without any seed. And in 98%, then the seeded fibril structure was acquired. The seeding does not only proliferate the fibril structure, at least of the main morphology, it also proliferates the proteolytic resistance or the proteolytic stability of the ex vivo fibrils. If you subject the fibrils from the mouse tissue to proteinase K digestion, find that the fibril protein persists for up to 2 hours under the conditions of our experiments. So you can see that the amount of proteinase K is actually quite substantial. For the unseeded in vitro fibril, the fibrils become quite rapidly degraded. And in the case of the seeded fibrils, there's at least some proteolytic stability until the end of the experiments. Now, that actually prompted us to look at the case of proteolytic stability a little bit more in detail, and we examined ex vivo fibrils of many different types: human AA fibrils, mouse AA fibrils, transdiretine fibrils with different mutations, immunoglobulin light chain from different AL patients, and in all cases, the fibril proteins are relatively resistant to proteolysis until the end of the experiment, which was 60 minutes in this case. By contrast, when we looked at in vitro formed fibrils from human SAA, murine SAA, transthyretin, a light chain fragment, human lysozyme, a beta peptide, alpha-synuclein, or human insulin, these fibrils were degraded quite rapidly, and there was very little proteolytic stability. So if I may summarize that first, out of the spectrum of fiber structures that can be formed by a given polypeptide chain, only some are found inside the body. The ex vivo amyloid fibers are highly proteolytically resistant, while the in vitro formed fibers are often much more sensitive to proteolytic digestion. And consistent fiber morphologies can be found at different deposition sites within the body and in different patients or animals, as long as they have the same clinical disease variant and express the same variant of the fiber precursor protein. So all of that together then prompted us to suggest a proteolytic selection mechanism, according to which a given fiber precursor protein is able to form a range of fiber structures, but most of them or many of them are not very stable, at least proteolytically, and can be readily removed by endogenous mechanisms of clearance. In some cases, however, it happens that a very protease stable fiber nucleus survives this hostile environment, is able to proliferate and form a fiber deposit, which ultimately then causes organ damage. Now, that mechanism is, in my opinion, also interesting in the context of what I would like perhaps to propose here as the Dobson Paradox. And I might come back to some of the old work that I did in the lab of the late Chris Dobson, together with others, also Fabrizio Chiti and others need to be mentioned, where it was suggested that amyloid fibers could be a generic state of the polypeptide chain, and that more or less any polypeptide chain is able to form amyloid. And the evidence in favor of that was that you can take many proteins that are either synthetic or not related to any disease. You incubate them under appropriate conditions inside the test tube, and you would find amyloid structures. And nowadays, the literature is full of hundreds and hundreds of other examples. Now, the Dobson paradox is basically, if it is a generic state of the polypeptide chain to form amyloid, why are only few proteins associated with amyloidosis? Depending on the definition, you have some 30 to 50 different sequences that are associated with disease. There are a couple of factors that can already be mentioned. On the one hand is protein concentration. You need to have a relatively high concentration of a particular protein to form aggregates. If you have an efficient folding reaction, then this antagonizes fiber formation, and you need to have appropriate conditions or kinetics for a particular sequence. Now, based on our findings on proteolytic stability, perhaps another factor can be added, and that is the ability to form protease stable amyloid fibers. So while the possibility to form any type of cross-beta sheet structure might be generic for polypeptide chains, the ability to form a protease stable amyloid fibers might be actually quite some sequence dependent. And similar also in the protein folding field, there are many different protein sequences, but only some are able to fold into a compact three-dimensional structure, and others give rise only to an intrinsically unfolded state. And indeed, when we analyze a number of these in vitro fibers that are not really related to any amyloid disease as such, then we also find that these are typically degraded under our proteolytic conditions. Interestingly, not in all cases. Here in alpha-lactalbumin, we also found some appreciable proteolytic stability. With that, I would actually now like to come for the last few minutes to the second topic, and systemic AL amyloidosis. It was also mentioned by band, systemic AL amyloidosis arises from the misfolding of immunoglobulin light chain. The basic clinical condition is a plasma cell dyscrasia, which gives rise then to high levels of free light chains in the blood. And in a fraction of the patients which are affected by these high serum free light chains, amyloid becomes deposited in different tissues, particularly in heart, kidney, soft tissues. Systemic AL amyloidosis is a relatively abundant systemic amyloidosis and a very aggressive one. If you have a heart involvement and there's no treatment, then the patients have an average survival of 50% in the first 6 months after diagnosis. Treatment is lowering the levels of serum-free light chains, for example, by chemotherapy. And in those cases where the heart is full of amyloid and does not work anymore, then heart transplantations might be necessary. Which is actually then sort of good for us because it gives us then an organ which is full of amyloid, so we can extract many fibrils. And this cardiac AL amyloidosis is also a case where the fibrils have a pathogenic relevance. In panel A, you see a tissue section from heart tissue, and the regions which are marked with a blue asterisk. These are infiltrations of the amyloid that completely disrupt the ordered structure of the cardiomyocyte tissue. And it is evident that this heart cannot work anymore properly because of the massive depositions of amyloid in there. We have now solved structures from AL amyloid fibrils from several patients. There's also one structure in the bottom right corner, which was solved by some groups in Italy. And the important feature of systemic AL amyloidosis is that it is a patient-specific disease. So far, there are now Cryo-EM structures available from 4 different patients, and the structure is different every time. The basis for this difference is, of course, because we have here immunoglobulin light chains, which are supposed to form parts of antibodies. So we have hypervariable proteins, which are on the one hand constructed by different germline segments. But there are then on top of these germline segments also mutational changes that are inserted mainly by somatic hypermutation, and they add then additional complexity. And for long it was thought that these mutational changes are also responsible for whether a light chain produces AL amyloidosis or not. But some of the details are not really understood very well. What we can now say, based on the analysis of the structure and the location of the mutations in the structure, the mutations occur all over the place. They are not necessarily only confined to the ordered fibril core. They also occur in flexible and structurally disordered regions. But all fibrils within the core, at least, are compatible with the specific fibril morphology which we see in this patient. In the case of the native globular conformation, it appears that the mutational changes occur in 2 regions of the protein. Those in the complementary determining regions in the CDRs, which are labeled here in blue, occur at the antigen binding site. That is actually not so super surprising because the CDRs form the antigen binding site. But those in the framework, which are labeled in pink, although they are distributed quite much across the sequence, they all seem to end up or seem to be preferentially ending up in a region, in a second region at the upper edge of the structure, close to the C-terminal end of the immunoglobulin variable domain. The structure in the fibril differs significantly from the globular fibril protein fold. The native immunoglobulin light chain, or at least the variable domain, which is the part which forms the fibril core structure, is in a native light chain a globular protein structure, while in the fibril, the conformation is relatively extended and flat. The interesting detail is retained, and that is the disulfide bond, which is present both in the fibril and in the native state. And if we zoom into this disulfide bond, there is an interesting difference between the native state and the fibril, because in the native state, it connects two segments which have a parallel N to C orientation. And in the fibril state, this is just flipped. This is rotated by 180 degrees, and that is true for all fibril structures that have been found, ex vivo fibril structures that have been found so far. And I think Fandrich has similar data for an in vitro fibril from a light chain. So we suggested that a rotational switch around the disulfide bond could be a key step in the misfolding of the immunoglobulin light chain, at least when you start from a natively folded state. And this conformational arrangement is so dramatic that it is not really possible to imagine it without a complete unfolding reaction of the native state to then form conformation of the molecule that is relatively flat and able to stack up in a linear structure of a fibril. The different fibril structures in systemic amyloidosis have also a number of fibril-specific features. For example, in some cases, we have evidence of internal cavities, like here in this fibril structure. Cavities A and B seem to be water-filled because they are lined by polar and ionic residues. There is another cavity which is lined by hydrophobic residues, and there is some density in there, suggesting that this is a molecular inclusion, probably some kind of a lipid. In another case, we found evidence that there are slightly different fibril protein structures at different Z-axial segments of the fibril, associated with structural breaks at the interface of these segments, or that there are cross-beta sheets attached on the outside of the fibril. They fit well with a polypeptide backbone. It is clear that none of the regions of the fibril protein, which we do not see in our Cryo-EM structure because they are conformationally disordered, these are labeled here in gray, is long enough to actually reach this position. So we don't know exactly what it is. It is probably an external cross-beta sheet attached, but we don't have much structural information at the moment about its identity. In one case, we had a fibril which is glycosylated. The glycosylation comes off here at residue 17. In addition to this carbohydrate, there's the disulfide bond, and the protein is partially pyroglutamylated, at least in those cases where the N-terminus has not been proteolytically truncated. The glycan chain occurs on the surface of the fibril and also on the surface of the native structure, as suggested by the inspection of homology models. But interestingly, this glycosylation does not necessarily accelerate fibril formation. If we refold the fibril protein from the tissue and then let it aggregate either in a glycosylated or in a deglycosylated form, then we find that the deglycosylated protein rapidly aggregates. These are the curves here in blue, while the glycosylated protein aggregates as well, but much more slowly. Both in vitro fibrils are not very proteolytically stable. The ex vivo fibril is highly stable to proteolysis. Of course, this condition is not very severe, only up to 5 minutes, but that is already sufficient to completely digest the in vitro formed fibrils, no matter whether they are glycosylated or deglycosylated. So perhaps one of the most interesting things that we found about this glycosylation is that it might affect the fiber structure, because when we look at the N and C terminal ends that are here below the disulfide, then they are spread apart in the glycosylated fiber, while in all other light chain fibers, they seem to be aligned. So they are in proximity, a little bit like a zipper, but in the glycosylated fiber, these two segments are spread apart. And actually here, this position 17, which arises from a mutation in this protein, which generates the glycosylation site, it is this residue which is glycosylated, and therefore the glycosylation sits exactly in this region of the structure which normally anneals in the other fiber structures, suggesting that the post-translational modifications along with the mutations affect the fiber morphology and help to generate a fiber morphology that is perhaps particularly stable to proteolysis and therefore pathogenically relevant. So I would like to finish by summarizing a couple of perhaps more general conclusions, what we think that we've learned about the way how amyloid fibers are structured, at least ex vivo amyloid fibers. So in all cases that we've looked at, the fiber cross beta sheets have a parallel beta sheet structure. The sheets can be left-hand or right-hand twisted. The fibrils consist of one or several fragments from the same precursor protein. Actually, there are no data that I showed you in that respect, but that several fragments are relevant for transthyretin. Amyloid fibers consist of one or more protofilaments, and they can contain a corona of other components or molecular inclusions which are integrated into the structure. The native fold of the fiber precursor protein differs substantially from the fiber protein fold, suggesting that it must become unfolded in order to form these fibrils. There are cases in which proteolysis precedes fiber formation and vice versa. And in vivo formed fibrils are polymorphic, but consistent fiber morphologies can occur at different deposition sites of one patient, or in different patients or animals that have a corresponding genotype and clinical phenotype. And ex vivo amyloid fibers usually differ in structure and proteolytic stability from the fibers formed inside the test tube. Amyloidosis is associated with, if not caused by, the accumulation of specific fiber morphologies in a patient or in an animal. And pathogenic amyloid fibers may be proteolytically selected. I would like to thank, on the one hand, a number of team members who contributed to the work that I presented in the slides, Giada, Christian, Thomas, Sarah, Lynn, and Matthias, as well as several previous members like Karik, Akanksha, Leonie, Marius, Falk, Mattis, and Jonathan. I also thank a number of collaborators, and in particular the German Research Council or Deutsche Forschungsgemeinschaft for funding. Thank you for your attention, and I'm looking forward to questions.
Yeah, thanks a lot, Markus, for this very exciting talk. There are already quite a few questions in the question and answers folder. So Sherris had two questions. So let me read them off here from the Q&A. Markus, could you comment on the presence of additional densities at the surface of ex vivo AA amyloidosis filaments compared to their seeded counterparts from recombinant protein? This is the first half.
Actually, when you look at we actually hoped or expected that the seeded fibrils would only contain very little of this density. However, we also see quite a bit of this diffuse density on the surface of the in vitro formed fibrils as well after seeding. Also in cases where we looked at in vitro fiber structures, for example, unseeded SAA fiber, we also had a bit of that density around. At the moment, we don't know exactly in all cases what it is. Initially, we thought that it is perhaps these are some other agents from the tissue. We certainly don't want to exclude that also part of the density comes from that. But as a matter of fact, I don't have a very specific case where I would say this density comes from this or that agent.
And with the second part of this question is, could it be that cofactors on the outside of the ex vivo filaments, which may be not present on the seeded filaments in vitro, provide the observed resistance to proteinase K?
I mean, it is possible that not all of the protease resistance comes from the fiber protein fold, and that additional factors which are carried over from the ex vivo extracts contribute to that. I think at the moment we cannot exclude that.
And the second question, also from Sjord, is what is the highest resolution Cryo-EM map for a right-handed twisted amyloid that you have? Can you see the main chain oxygen in it?
I have to think what is the highest resolution. Probably it is around 1.6 or 2.6 or 2.7 angstroms. We have not really tried to push it. Perhaps with one fiber structure like the AA ex vivo fiber structure, it's possible to push it a little bit further, but we have not tried it so far. But so far we have not seen this oxygen.
Okay, one question in the chat by David Eisenberg is, Markus, most interesting, from analysis of your extensive studies, can you formulate a general statement as to what structural features of amyloid fibrils enable a given seed fibril to induce fibril formation of a given recipient protein? It's a maybe a bit complicated question, and maybe you have to ask read it from the chat again.
I mean, it's hard to answer that, but I mean, I think that at least when it comes to the formation of the fiber structures that we see purified from the tissue, that there is a significant contribution also of certain side chain interactions. But at the moment, I have to admit, we don't have a very good understanding actually why the fibers look really the way how we observe them by experiment. And I would assume that one needs more or a better understanding what induces the formation of these specific fiber morphologies that we observe to more fully answer this question. Perhaps you have an idea. I mean, you look quite a lot at the interactions of the residues, in particular in the cross-section.
Okay, the next question comes from Martin Muschel. Great talk. So your results seem to support the notion that there's a fundamental difference between fibril-mediated systemic amyloid diseases versus oligo-mediated neurodegenerative amyloid diseases. Jeff Kelly, though, did present data suggesting that the treatment of TTR with tafamidis didn't noticeably reduce fibril loads in the heart, but significantly relieved patients' symptoms. Can you comment on that?
I don't think that all of the pathology in systemic amyloidosis only comes from fibrils. And personally, I find it also very hard to believe that all pathology in the brain comes only from oligomers, and fibrils are simply innocent bystanders and have nothing to do with disease. I think that there is a contribution of different types of aggregates in probably many, if not all, amyloid diseases. And for example, in systemic AL amyloidosis, there is also evidence for a contribution beyond the fibrils. And that evidence comes if patients are subjected to chemotherapy, which removes the circulating light chain in the blood. These patients feel better, and also many of the clinical parameters of these patients becomes better. The chemotherapy does not really change the amount of amyloid within the heart or in other organs, or not immediately, but the patients immediately feel better. However, even if you remove then the circulating light chains in the respective patients, the heart is still full of amyloid. And these patients then also have to undergo heart transplantation in addition to the chemotherapy, because in this case, really the amyloid is the problem. But I don't think that there is a completely black and white situation in neurodegeneration, only oligomers, and in systemic amyloidosis, only fibrils are pathogenic agents.
Okay. So Ashutosh Tiwari has another question. Have you tested fibrillation to alternate structure formation also in addition? Jumped the question. Have you tested fibrillation of the light chain after reducing the disulfide bonds? Does it change its aggregation kinetics?
I would very much think that it changes the aggregation kinetics and probably also the adopted fibril morphology, but we haven't tested that.
And another question in this context also from Ashutosh Tiwari is, does glycosylation lead to alternate structure also in addition to change in aggregation kinetics?
The structures which we got from that particular fibril protein, FOR1001 fibril protein, which we refolded from the heart, the fibril morphology that we obtained inside the test tube is more protofibril-like and not really looking like a mature and long and very regular fibril. And that was not really different, no matter whether we had the glycosylated or deglycosylated form. So perhaps there are some subtle structural changes that have escaped our analysis, or that were not really visible by electron microscopy. But so far we did not detect any changes, but it's possible that there are.
One related question from Markus Zweckstetter. Fantastic work and presentation, Markus. Could you speculate on what could be the reason for increased proteinase K stability of ex vivo fibrils? How about PTMs interfering with PK digestion?
Well, in many cases, the fibrils are not really substantially post-translationally modified. In systemic AL amyloidosis, there are, the disulfide bond of the immunoglobulin light chain. There's also a little bit of pyroglutamate because in many cases, light chains have a glutamine residue at the N-terminus in their native form. And if the N-terminus is not truncated, which is actually the case in many fibril proteins, then you also find a pyroglutamate. And the glycosylation occurs only in some 5% of the patients. So it is not a very abundant modification. In systemic AA amyloidosis, I don't think that we have a lot of evidence that the mouse or the human fibril protein is really substantially modified post-translationally, except from the proteolytic truncation. Proteolytic truncation is something that we find in many cases. But in many cases that we looked at so far, there is not so much post-translational modification, but that doesn't exclude that in some cases it is important.
And the second question, could you again explain how to determine the right and left-handed twist?
We actually have two methods, either scanning electron microscopy or transmission electron microscopy. And in both cases, we do a platinum side shadowing, which you can compare to snow falling on a landscape. So you cover then your structure with this platinum. And if you look at it by, in particular, scanning electron microscopy, which is quite convenient, then you can determine the handedness, whether it is right-handed or left-hand twisted. At least in those cases where the cross-section of the fibril is rather anisotropic. If you have a cross-section which is circular, and therefore there are no height variations of the fibril on the grid, then this side shadowing will also not reveal many differences. And then it's very hard and in some cases even impossible to determine the twist.
I could imagine that Marcus maybe was referring to this 5-psi Ramachandran diagram that you showed, which had a diagonal, and then above below the diagonal you had this left and right-handed twist. So you related this somehow then to the secondary, to the structure.
Yeah, I mean that is actually some very old stuff, and I think it was Cyrus Chosia who initially noticed that. I think in 1973 or so, that in globular proteins, which usually have a left-hand twisted sheet, at least if you define the twist in a certain way, then you have more psi-phi pairs on the right-hand side of the diagonal than on the left-hand side of the diagonal. And if you look at the molecular structure of a polypeptide chain, then psi-phi pairs on the right side of the diagonal induce a helicity of the polypeptide backbone. And I think that is also part of the reason why Sjors asked for the carbonyl oxygens. And you then have a helicity of the polypeptide backbone, so that if you add several of these polypeptide chains together to form then hydrogen bonds and a beta sheet, you end up with a left-hand twisted beta sheet, as in globular proteins. And in those cases where we looked at the psi-phi distribution in left-hand twisted amyloid fibers, there's also the majority of psi-phi pairs on the right-hand side of the diagonal, as expected. And in those cases where we have a left-hand twist, we have more psi-phi pairs on the left-hand side. I hope I did not confuse it because I sometimes do, but sort of it tries to relate the handedness of the structure to the model that we then generated based on the 3D map.
Okay, another question from Danilo Miladi. Thanks, Marcus. Amazing talk. 2 questions. In the absence of cofactors or helper proteins, is transmissibility linked to amyloid fiber structure, and to what extent?
Good question. I hope I can give you an answer in some months.
Okay. Then second question, is proteolytic stability of ex vivo seeded fibrils related to the extent of beta sheet hydrophobic structure ratio, so the beta sheet to hydrophobic structure ratio exposed to the solvent? And
that is now seeding, whether seeding ability is correlated to that or not.
As the proteolytic stability of an ex vivo seeded fibril, of a seeded fiber related to the extent of the ratio beta sheet hydrophobic structure exposed to solvent.
Actually, interesting question, but we should look at that. No, perhaps, but I don't know at the moment. Yeah.
So another question comes from Min Su. Were the cryo-EM structures determined by a single particle approach or the combination of both SPA and helical reconstruction?
We used for the reconstruction in recent times almost exclusively the Relion program by Sjors Shear's group, which is basically then a helical particle reconstruction method, basically implemented in a single particle framework. I hope that answers it.
Another question comes from Christine Pohl. Thanks for the great talk. Do you have an idea which structural features makes a fibril proteinase K resistant?
Yeah, I think we had a similar question before. I wish I would, but at the moment I cannot really answer.
There's another question from an anonymous member of the audience. Is there any suspected mechanism for exclusion of R1 residue in human SAA amyloid formation?
What is known that a fraction of the SAA protein which circulates in the blood is disarginated. So you do find it in an acute phase response that also the SAA which you can isolate from the blood is partially disarginated. So I assume there is some kind of protease or at least an aminopeptidase that is able to remove this arginine, but I'm not aware of any specific factor that has so far been identified.
The next question is from Astrid Greislund, I think. Do you think that the intermediate oligomeric states are also different and therefore give rise to the different fibrils? And are other components present in the fibrils like lipids or metal ions?
I mean, if you grow a fibril, then the most likely mechanism is that you have initially a seed or a small part of the fibril or a fragment of the fibril that already has the structure as in the final fibril, and that this then grows out and extends into a linear structure. So I would expect that in those intermediate aggregates or oligomers that are on pathway to a specific fiber morphology, you already have nascent elements of this fiber structure present. However, I would assume that at the oligomeric state, probably a large number of structures are sampled, and there are from several groups, there is evidence that in these early oligomeric states that there is a certain extent of antiparallel structure as well. We also found that many years back for A beta, while in the terminal fibril state, at least at the time only neutral fibril states were known, it's then parallel beta. So I assume that in these early intermediates, a lot of different possibilities for a polypeptide chain to assemble are explored, and that only some are then really productive to give rise to a fibril.
Okay, then one more question from Carmelo La Rosa. Hi, Marcus, very interesting talk. It is interesting to me or to see that you have observed phospholipids trapped in the cavities formed in the fibrils generated in amyloidosis. Have you observed this in other types of fibrils as well?
First of all, greetings to Catania. It is actually only for one patient in systemic AL amyloidosis where we have some evidence that looks like a lipid inclusion, but there are additional cases for lipid inclusions in the labs of Shears, for example, that there are inclusions in alpha-synuclein fibrils. So it is an observation that you can find a couple of times meanwhile in the literature. But in our case, we only have such a lipid in or such an apolar, probably apolar inclusion only in one case.
Okay, good. Now the longest question actually comes from Luis Del Pozo. According to the light chain amyloid aggregation mechanism suggested in your presentation, it is the fraction of fully unfolded molecules that fuels the fibril assembly reaction. Is this correct?
Okay, that was not so long. Louis,
the second half is still coming.
That was the first part. Okay. Yeah, well, I mean, if you look at the fiber structure, it is really so much different from the native state that you cannot really leave a part of the fiber of the native structure intact to form this fiber conformation. So I would expect that there basically all parts of the native state need to unfold. I do not know at the moment whether there is one complete unfolding event and then there is a full random coil which then associates, or whether the transition starts in one part of the molecule and then perhaps extends to other parts of the molecule.
Maybe I can read off the question, because Luis writes further that in some in vitro experiments, they showed that the correlation between the concentration of denaturant, such as urea or gunidinium, and the rate of fiber formation, both in fibrillogenesis from soluble protein and by seeding, is not linear. And instead, it has an inverted U shape with a vertex around the CM. And this suggests that partially folded species rather than fully unfolded monomers are the key players in the aggregation reaction. And how to reconcile your mechanism with that proposes that partially folded intermediate states play the key role in fibrillogenesis.
Yeah, no, I understood it, and I think that is absolutely valid. I mean, these kind of observations have been around for more than 20 years, I would say. Also during my PhD, then there was this evidence that if you denature the native conformation, that there are some intermediate denaturant concentrations under which then fiber formation occurs. And if you have very high concentrations of denaturants, you don't have fiber formation. And if you have very low concentrations of denaturants, you basically have the native state and you also don't have any fiber formation. Now, my explanation for these, for the observation that these intermediate concentrations of denaturants are promoting fiber formation is that with the denaturant, you want to you want to do something very delicate. On the one hand, you want to disrupt non-covalent interactions which are characteristic for the native state. But on the other hand, you don't want to disrupt the non-covalent interactions with the fiber form. So I would think that the reason why you see this optimum of intermediate denaturant concentrations is that this is sort of the balance of denaturant concentrations which are just sort of sufficiently destabilizing and unfolding the native state, but which are not so strong that they prevent the formation of amyloid fibers. Because at least in our hands, when we expose ex vivo or in vitro fibers to high concentrations of guanidine or urea or SDS, then they usually disassemble into monomers. So you would assume that very high concentrations of denaturants are not productive for fiber formation, because they denature both the fibers and the native state, while at low concentrations, the native state is stable and you don't see any fibers as well. So you would need these intermediate concentrations.
Okay, I think this was addressing the question quite nicely. And I think we also reached the end of the Q&As. Yeah, thanks, Markus, for this very nice discussion and for the presentation, and yeah, and I maybe turn back the stage to Rams.
Markus, great talk. Thank you very much for this nice talk.
Thank you. Pleasure.
So yes. A lot of questions nicely answered. So let's revisit this water channel that you see in systemic AL amyloids, but not in systemic AA amyloids. Am I correct?
I see it in certain patients,
yeah,
with AL amyloid, and I see it in the human AA fiber, but not in the mouse AA fiber.
So when do you see them? Like, do you have any ideas of when this water channel occurs, and what are their significance in stability, maybe proteolytic degradation, etc.?
There's not much that I can really answer to that. We do see it every now and then. I think in the case of the light chains, in that case where we actually find these water channels, that light chain has a very acidic patch. It has several acidic residues in a row. So it is actually a structural motif which you would never imagine in the core of a fibril. I mean, if you intuitively would build this fibril, then you would place this region of the structure on the outside, because you cannot imagine it to be buried in the core. But for some reason, it is buried in the core. And the only reason why it is tolerated there is that the structure builds these two water channels around this highly acidic motif, and therefore sort of helps to compensate it somehow.
Great. So you also mentioned about this 95% versus 5% polymorphs. Are there any factors that control the magnitude, the size of this population?
I would assume, but I cannot say much about it. I mean, one of the interesting features is that if we extract fibrils from mice, then 95% of the fibrils have 2 protofilaments and 5% have 3 protofilaments. When we use these fibrils extracts as seed in vitro, we have 98% of the fibrils with 2 protofilaments, and we completely lost the fibrils which have 3 protofilaments, or almost completely lost the fibrils which have 3 protofilaments. So in that case, I would assume that it is kinetically favorable to extend a fibril which only has 2 protofilaments. Simply is faster because you only need to extend 2 ends compared to a fibril which has 3 protofilaments. So perhaps that 3 protofilament fibril is then more stable, and therefore you see it in vivo situation where it has more time to accumulate. But these are relatively crude speculations at the moment.
Thank you very much. Are there any questions from Magda, Sam, or anybody else in the audience? Ben, yourself? Any questions?
No.
Yeah, I'm sorry. I just my Zoom still keep on playing tricks on me. I actually had a question about polymorphism, the triple protofibrils and the double protofibrils. I assume you were not able to follow on studying their toxicity and their properties. Do you think that they're more toxic or something special? And when you look at the outside, are they having different properties? You would speculate that they will have a different properties.
At the moment, it is not possible for us to make biochemical preparations which are pure with one of the fibromorphologies and pure for the other fibromorphologies, which you would probably need to study different biological effects. So there's not much that we can say about that at the moment. I mean, it's possible that they differ in certain biological properties, but we also have not tested it really at the moment.
And I have another question. It's about shearing of the fibrils. So when you're looking at the proteolysis, it's not necessarily that it's overlapping the proteolytic stability on stability with the ability of the fibrils to be sheared by mechanical forces. Can you comment on that? Do you see any differences, or have you checked that?
You mean whether the fibrils have different susceptibility to shear forces?
Yeah.
Yeah.
And whether this overlaps with their proteinase K stability.
No, we have not looked at that so far. At least we don't have much evidence that the ex vivo fibrils always have a high thermodynamic stability because both the ex vivo AA fibril and the in vitro AA fibril, they denature quite readily in higher or in medium guanidine concentration. So both of them don't seem to be super stable, really.
Okay. Thank you. I just had all my questions. Thank you so much. It was really, really inspiring talk.
Thank you. Pleasure. I'm sorry that I could not probably answer them better.
No.
So Rodrigo, there's one person raised their hand. Rodrigo Gallardo, would you like to ask a question? I can allow you to talk.
No, my question was already kind of already answered
Okay.
in the previous question. Thank you so much, and thank you so much again, Marcus, for the presentation.
Thank you.
Okay, so we are at the end of the presentation. Thank you very much, Marcus. Again, thank you very much, Ben.